All right, good afternoon, everyone, and thanks, James and Jay, for putting on a fantastic conference. We'll get to tea time here shortly, so hang in with me. Those of you who don't know me, my lab is at the University of Queensland, and we are working with human pluripotent stem cells to study as a model system for studying cardiovascular development and disease. And while most of my talk is going to orient around our work on the genomics approaches that we're using to study differentiation, Meredith illustrated the work that we're also doing in the area of drug discovery with a number of collaborators at the IMB, including Glenn King and others that are focusing on the incredible resource we have in the area of toxin and venom extracts to identify novel cardiovascular drugs. And I just want to illustrate some raw data that we've generated recently where we've applied acutely to these iPS-derived cardiomyocytes, extracts from consonale toxins. And you can see that there's just an incredible diversity of effects that these toxins have in the way of the behavior of the cardiomyocytes in this acute treatment. And this is a part of a broader scope of analysis that we're doing, including work with consonales as well as the box jellyfish, a species that will kill you in about 10 minutes. I told my Ph.D. student to wear two sets of gloves because I didn't want to call her parents. And then we've also got the cane toads and, of course, the spider venoms. And so this is, of course, something that taps into all of our interests in the way of using iPS derivatives for translational applications, whether they're for drug discovery or for understanding mechanisms of disease or cell therapeutics. But if we take a step back, ultimately all of this has to be grounded in developmental biology and our understanding of how to control cell fate decisions. And so as we try to understand the mechanisms that control the diversification of cell types from pluripotency, ultimately this has to be informed by our understanding of how the heart develops in the body. And it has now, over the last couple of decades, enabled us to give rise to a vast diversity of cell types that comprise the cardiovascular system and position us now to really make some exciting discoveries going forward that affect our ability to understand and contribute to therapeutic approaches for cardiovascular disease. So in just a study that we undertook with Joseph Powell's group at the IMB over the last couple of years, we wanted to understand the mechanisms that underlie cardiac differentiation at single-cell resolution. And while many of the mechanisms that we know underlie each of these fate choices, we wanted to study the subpopulation composition and the full transcriptional networks that underlie these stepwise transitions in differentiation. So to do this, we used a small molecule Wnt modulation protocol that is widely utilized, and we didn't introduce any enrichment steps just to be able to capture all of the diversity of cell types that are derived during this differentiation protocol. And so this is a t-SNE plots that reflect the more than 43,000 cells that we've captured, comprising stage-wise transitions from pluripotency to this gastrulation stage of differentiation, the progenitor cell, and then 2 stages of more committed cardiac derivatives. And we worked with Patrick Tam's spatiotemporal transcriptional data from the gastrulating mouse embryo, as well as Sean Wu's data in the analysis of heart development in vivo to identify the subpopulation identities across this diverse set of cell types that are comprised in the context of cardiac in vitro differentiation. So I just want to highlight a couple of key points that we have used in the context of this analysis. The first is actually implementing and devising computational strategies that allow us to move from big data to actual mechanisms. I don't think there's any shortage of information anymore now that we have these incredible capabilities of extracting transcriptional data at single-cell resolution. But how do we actually make meaningful information or mechanistic insights about these complex processes? So we've been making some headway in that space, and then utilizing these types of discoveries and strategies to identify molecular control points that control cardiac differentiation. So to begin with, one of the key questions that we want to understand was how do we map out the complex interactions and fate choices in a trajectory prediction approach to understand how cells navigate from pluripotency into the cardiac fate. And we implemented a number of analyses that use these pseudo-time type of trajectory predictions as well as the entropy-related strategies for predicting lineage trajectory across subpopulations at single-cell resolution. And these didn't work very well, largely because these strategies, the assumptions that are built into them, are not well suited for these types of complex time course differentiation data sets and the level of heterogeneity that we have in our populations. So we collaborated with Ziv Bar-Joseph's group at Carnegie Mellon University to adapt an approach called SCDIF that uses transcription factors and their regulatory networks to assign parental and descendant states in an unsupervised manner. And this was the key to enabling us to really identify the trajectory predictions that underlie the fate transitions during differentiation. And again, this really gets at the fact that we are focusing in on regulatory molecules, the transcription factors themselves, as that key determinant of being able to dissect out the complexity of these fate choices. And I want to highlight the first is that there is a diversity of cell types on this exit from pluripotency that comprise mesoderm, mesendoderm, and definitive endoderm. And we believe that this stoichiometry, which reflects normal development of the heart, is critical for being able to instruct a small proportion, only a third of the cells, that ultimately give rise to all of the derivatives in this differentiation protocol. The second is that we wanted to understand the mechanisms that are underlying the specification of cardiomyocytes in particular. How well are we accurately patterning the complex gene regulatory networks that underlie these, the derivation of cardiomyocytes? So we surveyed a vast diversity of transcription factors and epigenetic regulators that we know are important for heart development in vivo, and identified this gene HOPX, which probably many of you well know has a very long history in cardiac biology, is well known to be one of the most essential regulators of cardiovascular development. And we found, in the context of our differentiation, that this gene is not accurately activated as we would expect it to based on our understanding of the development of the heart. What we find is that this HOPX is activated in endothelial cells at day 5, but not in the cardiac precursor cells, as we would expect. Instead, it's literally 3 weeks after they've started to beat that we see transcriptional activation of HOPX, and in that case, we're only detecting it in about 16% of the cells, whereas in normal heart development, almost all cardiomyocytes are derived from a HOPX origin. So we set out to determine there must be some signal or cue that's missing in the context of differentiation that we need to address in order to accurately pattern transcription of this key cardiac regulatory gene. And so to not go into all of the details on this, we worked with Hannele Ruhila-Baker at University of Washington and using gain and loss of function models to be able to identify cardiac hypertrophy as the key signal that is required to transcriptionally activate HOPX. And so in the context of hypertrophic growth, we get a robust activation of HOPX. And you can see that it is ubiquitously expressed across this population. To understand the dependency of this hypertrophic response in HOPX transcription, we used a CRISPR-I cell line that was generated by Bruce Conklin's lab at the Gladstone Institute that allows us to functionally block these two transcriptional start site of HOPX. And the take-home point from this is that while HOPX is transcriptionally activated by hypertrophic growth, the hypertrophic genes, at least those that we surveyed, were not significantly differentially regulated by the loss of HOPX. Instead, what we found was that the cells failed to transition into a more definitive state during this differentiation process in the absence of HOPX. So they become essentially caught at just after specification and are unable to activate transcriptional networks as well as just morphogenetic changes in terms of their transition toward a more mature, definitive cell state. So this has led us to recognize that in these high-density monolayer differentiation protocols, the absence of hypertrophic stimulation is one of these key cues that's required for us to be able to accurately pattern in vitro-derived cardiomyocytes as they occur during embryonic development. All right, so the other aspect of this analysis was a focus on this non-contractile cell type that has been utilized and is commonly utilized in tissue engineering applications because it's a cell type that's required for remodeling the matrix and enabling these engineered tissues to form. When we analyzed the transcriptome of this population, what we found was that we got a significant enrichment of pathways, genes associated with vascular development. And so we took a closer look at this analysis, and if you'll just focus in on this particular subpopulation here, which are these non-contractile cells, what we found is they're not expressing the cardiomyocyte genes. They're also not expressing genes that are known to be involved in vascular angiogenesis like TAL1 and VE cadherin. Instead, we see this signature of genes that are associated with second heart field development, including the HOX genes, TBX18, PIDX2, FGF10, and other genes that would indicate that we're deriving some kind of a second heart field outflow tract derivative. We worked with Sean Wu's data to look at, because he had the spatial resolution of being able to look at different genes that are reflective of different compartments of the heart. If you look here at the outflow tract versus left ventricle in vivo, you can see that this is some of the most significantly differentially expressed genes that define the differences between these subpopulations. And similarly, our non-contractile cells versus the cardiomyocytes similarly show this type of differential expression that recapitulates the comparison of these two cell populations in vivo. And taking a more unsupervised approach to this, we analyzed the entire transcriptome of these different cell types relative to multiple cell types as they occur in vivo. And you can see that the cardiomyocytes are more strongly associated with the myocardium, as we would expect based on this Spearman-Rank correlation analysis. But when we look at the non-contractile cell types, comparing endothelium or fibroblasts or myocardium, it's really the outflow tract that is the most substantially correlated with this particular subpopulation that we're deriving. So this is setting us up. We're undertaking some future directions here to try to understand the nature of this cell population. Does it accurately reflect a second heart field type of cell type? And to what extent does that actually reflect in vivo development? All right, so just to wrap up, I want to bring to your attention a new strategy that we're utilizing in the lab to try and identify cell type-specific regulatory genes. And this has really been the key focus of my lab, taking the fact that we work with big data sets now with single-cell analysis, but how do we make sense of this in a mechanistic way to understand biological processes? And the problem that we struggle with a lot is the fact that we rely on some kind of external reference point like differential expression or on some kind of prior knowledge like network analysis to make inferences about the nature of mechanisms that underlie cell populations. But ultimately, these types of strategies are very limiting because they limit both novelty and scalability. And so I just want to introduce an approach that we've developed in the last couple of years called triage, which stands for Transcriptional Regulatory Inference and Analysis from Gene Expression. And the approach that we've taken is to recognize that we have an extraordinary power of information available to us through these consortium data sets like the NIH epigenome and the ENCODE data sets that allow us to now understand the complexity of epigenetic states, also in terms of their control of transcriptional activity across an extraordinary diversity of cell types. And so the way that we have approached this is to analyze all of the histone modifications from more than 100 different cell types from the NIH epigenome. And the analysis that we've done has really focused us on this H3K27 trimethylation as a key domain or histone modification that we can use as a quantitative approach for being able to prioritize cell type-specific regulatory genes across the genome in an unsupervised manner. And so we've devised what we're calling a repressive tendency score that allows us to make a prediction about every gene's likelihood of being a regulatory driver of cell identity. And while we have this ability to make these predictions genome-wide, the way that we're implementing this at a scalable level is to recognize that we can take now any gene expression data set from any cell type as a unique signature of a cell's state. And by imposing our prediction algorithm, we can now infer the regulatory drivers of that particular cell state and understand now the development, the regulatory drivers of development, of disease, of cell reprogramming, and it really opens up the door to analyze mechanistic data from these types of large data sets. So let me illustrate how this works. So because we all love the heart, I decided I like it too, and this is just focusing in on a cardiomyocyte sample, just transcriptional readout of a cardiomyocyte. So if you were to rank these genes on the basis of overall expression, what you would see is that among the most highly ranked genes of a cardiomyocyte are the sarcomere genes and housekeeping genes, as we would expect, because these are the genes that are in high abundance. But ultimately, this is just a descriptor of a cardiomyocyte, and we all want to know what is the driver of the cell's identity. So by introducing this triage analysis without any other reference information in regard to the fact that this is a cardiomyocyte sample, what this does is that instead of having this rank order of high abundance, these genes are now flattened down, and you see popping up down the list these genes that seem to have gained a higher prioritization. And these genes are the core drivers of cardiomyocyte identity. So now, instead of structural genes and housekeeping genes at the top, we now have the core driver genes of cardiomyocytes, including NKX2.5, GATA4, the hand factors, IRX4. And this is now enabling us to identify the regulatory identity of a cardiomyocyte. Now, we've applied this across the entire genes by cells matrix of our in vitro differentiation data set to look at the ability to pick up the regulatory drivers of the stepwise transitions in cardiac differentiation. And I'm highlighting here gene ontologies that are strongly enriched for transcription factors and signaling molecules that underlie these fate transitions, in contrast to gene ontologies that are associated with just structural genes or housekeeping genes. Now, if you were to just look at transcriptional expression data across this time course, you can see that those that are most highly ranked are typically just structural housekeeping-related genes. But when we analyze this data now with triage, we can now pick up all of the, or many of the key regulatory drivers that underlie stage-wise specific transitions in cardiac differentiation throughout this broad diversity of cell types that are reflected in our data set. And I don't have time now to go into the details of this, but I want to illustrate the fact that we are working with people from Europe and North America and around the world to try to utilize this to address some very big questions, trying to understand mechanisms of disease, not only cardiovascular disease, but cancer as well, understanding developmental biology, and it's not just about human developmental biology. We're working with Lionel Christian at New York University to look at the regulatory drivers of the invertebrate tunicate and the heart specification mechanisms that underlie that model system. So there's broad diversity and application for this in the context of both developmental biology and disease, as well as novel gene discovery. So with that, I want to just close by highlighting the fact that this brings us back to the importance of utilizing pluripotent stem cells really relies on our ability to gain mechanistic information about the nature of these cells and being able to control their identity in order to position us to both undergo studies in novel gene discovery, understand how to specify the broad diversity of cell types that comprise the complexity of the heart, and ultimately positioning us to discover novel drugs or develop cell therapeutics that have the ability to transform the future of medicine. So with that, I just want to thank many people that have participated in a variety of these projects, including Joseph Powell's lab, who's currently at the Garvin Institute. Mikhail Bodin is a key collaborator on the triage analysis tool, the chemist at the IMB in terms of our drug discovery, and then my lab who have been instrumental in being able to push these projects forward. So with that, I'll take any questions.
If there are no burning questions, we might just wrap up the session there.
That sounds great.
Head off to tea. Break. Join me and Asa in thanking all of our speakers for a great session. We'll be back here just before 3.